which is learning and the brain. We're going to talk in this podcast today about the human nervous system. We will also talk about brain structures and functions, including neuroimaging. Parts of the brain, we'll talk about lobes of the brain, the hemispheres, and then we'll get into talking about development of the brain and factors that influence that brain development. We'll kind of wrap up the lecture by talking about how pre-wired we are and maybe even get into some educational implications. So the diagram you're looking at here is a diagram of the human nervous system. And that pink part, which makes up the brain and spinal cord, that is our central nervous system. The blue part is your peripheral nervous system. So that central nervous system, like I said, involving the brain and the spinal cord, that is what connects everything we're experiencing with how we react and what we do. The peripheral nervous system is kind of considered our messenger system, and this is where our autonomic nervous system is housed. So within that autonomic nervous system, we have something called the sympathetic system, and that's what handles our fight or flight response, which tends to be when we're in anxiety-provoking situations, and the parasympathetic nervous system, and that's what handles kind of our day-to-day activities that keep us alive, so things like resting and digesting or feeding and breathing. What you see here is a diagram of a neuron. Now notice that those points where the axon of a previous neuron kind of ends, and you see those axon tips and kind of those axon tips coming into contact with the dendrites of the next neuron, they're not actually touching, but rather they're pretty close to one another, and there's a little bit of a gap there that chemicals are transmitted across, and that's what sends messages from one neuron to the other. So essentially what happens is messages come from the soma, which is kind of the cell body here in our diagram. They travel down the axon. It's an electrical signal that is called action potential. We'll talk about that in a second, to the dendrites. And then the messages cross there at the synapses. So let me show you this video on action potential. We'll see if we can get this to work. That will better explain the electrical signal that gets carried down the axon, kind of triggering this process. Action potential. And what's really important for you to understand is not so much the chemicals or anything like that involved in this, but I just want you to kind of see the process by which the kind of initial communication happens between neurons.
Not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal, called an action potential, that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
All right, so you get a pretty good sense of the action potential process and what that looks like. And now what we're going to do is kind of switch gears, and you've seen how the action potential travels down the neuron. Now we're going to actually look at how these neurotransmitters are released across the synapse to communicate these messages between neurons. Take a look at this video on how neurotransmitters work.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
All right, and so now you're kind of able to see what that action potential actually triggers, right? These chemicals are released from one neuron, they cross that synapse, and they are taken in by the other neuron. And this is where things like SSRIs, which are those serotonin reuptake inhibitors, those things are acting on these neurotransmitters. So let's continue to kind of talk a little bit more about neurotransmitters and what they do. Now, this is not an exhaustive list of neurotransmitters, but I want you to get a sense of how each of these things play a role in learning. So we have glutamate, and that is implicated in memory storage. So when your glutamate is too high, this tends to lead to things like seizures, strokes, Huntington's, Parkinson's, Alzheimer's, all things that kind of your memory can be impacted by. Dopamine is something that we typically associate a lot with motor behavior and our reward system. So when your dopamine is too low, this can lead to things like Parkinson's, and when it's too high, this can lead to things like schizophrenia. Serotonin, you guys all probably are fairly familiar with that, and that relates to those SSRIs, again, that are used typically to treat depression. So the idea here is that when you have low serotonin, then you end up experiencing depression. Serotonin often regulates things like sleep, appetite, memory, learning, mood, and even temperature. And you can see that all of these things are impacted when you're experiencing depression. You tend to either sleep too much or not sleep enough. Appetite is either too much, too little. There's actually a lot of research now to show that when people are experiencing depression, their IQ is actually lowered, their ability to remember and learn things is actually lowered. So again, huge, huge impact on learning here. Acetylcholine, this one, this neurotransmitter is often implicated in things like arousal, learning, our motor system, or short-term memory. And then finally, GABA. So GABA, when you have low levels of GABA, this is often associated with anxiety. And then the other thing that's interesting about GABA is that over time, as we age and as hormones kind of flood our brain in puberty, GABA changes from excitatory to actually having an inhibitory effect. So one of the things you notice here is that there's a lot of overlap in the role between these neurotransmitters, and typically speaking, when we're talking or thinking about disorders, there's not necessarily just one neurotransmitter that's involved. So anxiety is a great example of that. We think that anxiety tends to be related to imbalances in both serotonin and our GABA levels. So again, all of these neurotransmitters kind of function in different ways, but you can see the many impacts they have on your learning. So how do we take a look at the brain structures to kind of see if they're working or functioning in the way we need them to? Well, we use neuroimaging, and one of the first kinds of neuroimaging we can do is called functional neuroimaging. So this is where we're looking at changes in blood flow or electrical currents. So a lot of times we will do things like activation studies where we will have someone in either kind of an fMRI, an EEG, or a SPECT, and we will then put different stimuli in front of them to see what activates in the brain. And so again, this is looking at either blood flow or electrical currents activation. Structural neuroimaging, not surprisingly, is often used to give us a detailed picture of the brain. And it's oftentimes helpful in detecting a traumatic brain injury someone might have experienced as a result of a car accident or even drowning, those types of things. So ways in which we can do structural neuroimaging include MRIs, CT scans, and this is just one example. So you can see here on the left the structure of a typically developing 3-year-old child's brain who's been in a kind of nurturing environment, as opposed to the brain of a child who has been in an extremely neglectful environment. So you can see that left brain is kind of lush, it's thick, it's got tons of gray matter there, lots of connections, and in the more impoverished environment, the child's brain certainly has developed quite differently. So now we're going to talk about the different parts of the brain. And typically when we're talking about the parts of the brain, we're thinking of either the hindbrain, which is kind of the base part of the brain there, your brain stem, including the medulla, the pons, and the cerebellum. And this is the part that regulates all of our basic physiological processes. So things like breathing, sleeping, heart rate, all of the things that keep us alive. So an injury to the hindbrain can be deadly. This midbrain area, this is where the reticular activation system is stored. And one of the big jobs for this part of the brain is to coordinate our vision and our hearing. And it also helps us to regulate attention and consciousness. Now, the part of the brain that we're going to kind of focus on the most here in this lecture is the forebrain. And this is where all of the lobes are stored, as well as the cerebral cortex. So this is really the part of the brain that's handling all of our complex mental processes, including things like learning. So we can break up this forebrain into 4 separate lobes. I'm going to start by talking about the frontal lobe. So the frontal lobe is really where our executive functioning powerhouse is. And executive functioning is made up of things like attention, the ability to inhibit yourself, problem solving, reasoning, being able to plan for things like long-term projects. And your frontal lobe is not actually fully developed for men until their mid-20s. For women, it's more like 18, early 20s. So you might notice differences in dating around that age, and that might help to explain some of it. So the deal with frontal lobes is when you think about someone like a teenager or even a young child, as those frontal lobes develop, their attention, all of those things, their ability to inhibit themselves are expanding. But when you have a really, really young child, oftentimes their frontal lobe is very underdeveloped, right? So you'll notice that they have very short attention spans. They often say things that they probably shouldn't be telling people, but they really don't have the ability to regulate their inhibition or regulate their thoughts yet, right? Inhibit themselves. Problem solving can often be really tough for these kids, right? You see tantrums a lot. So all of these things relate back to frontal lobe development. And we have to keep this in mind when we're doing things like giving kids directions, because they might not have the attention span to handle 2 or 3 step directions, right? So knowing brain development is essential. The parietal lobes, that's this yellow area. This parietal lobe handles our somatosensory cortex. It's also involved with attention and processing, as well as spatial planning. So things like knowing how to efficiently organize your dishwasher, depending on what kinds of dishes you have, would be spatial planning. The occipital lobe is that pink lobe there at the back, and that's what handles your vision and visual memory. And then finally, the temporal lobe, that green area there. This is what handles your language. So your Broca areas are here, and your Wernicke areas are here. And this is where auditory information is processed, and to a certain extent, long-term memory is also processed. So again, there's some overlapping between the functions of each of these lobes, but kind of good to know what each lobe handles, especially if you were to encounter someone who had experienced a brain injury, for example. So this idea of left and right hemispheres and having different functions is actually a little bit dated. There's now some research to suggest that there really is not this kind of division of I guess, traits across the hemispheres. But we're going to talk about what was a long-held belief, which is that this left hemisphere, which is connected to the right hemisphere by this corpus callosum, the left hemisphere is really responsible for logic, math, language, reading, writing, and analysis, and then the right hemisphere is more in charge of personality, creativity, intuition, music, art, spatial abilities. You probably already know that each of the lobes control the opposite side of the body. But again, like I said, there's a lot of controversy about this idea that the left hemisphere kind of handles these areas of our skills versus the right hemisphere. So just kind of keeping that in mind. All right, now we're going to talk about development of the brain. So when, as babies are developing in the womb, prenatally, what we see is that the brain begins to develop as kind of a tube after about 25 days into conception, after conception. So at this point in time, approximately 50,000 to 100,000 neurons are growing per second. In the second trimester, all of these neurons begin to migrate, and this is when those dendrites begin to grow. So the ones that don't kind of make connections with another neuron will then die off. So after the baby is born, between the age of birth all the way up to 3 years old, the brain actually triples in size. And what's going on here are several processes. So you have synaptogenesis, which is where synapses are formed, and this is increasing dramatically after birth. You also have differentiation, which is where the synapses begin to kind of take on their own functions. And then finally, as this baby, as this infant begins to encounter more and more things in their environment, they start weeding out synapses that aren't really being used anymore. And this is, like I said, largely based on their experiences in the environment. And the big idea here is that, the more synapses you have is not necessarily the better, right? We want to make the brain efficient. And so as babies are interacting with their environment, these kind of paths within the brain begin to develop and things that aren't needed are cut out. Finally, myelination occurs, and this is where those neurons get that kind of fatty coating along the axon that help to make the action potential, that communication down the neuron, down the cell, more efficient. And this is, again, within the first few years after birth. So as we age into middle childhood, adolescence, and even into adulthood, pruning continues. I've already mentioned that the frontal lobes aren't fully mature until late adolescence or early adulthood. So kind of think about that, right? I mean, teenagers start driving at 16 and they don't even have a full frontal lobe yet. I can think of how many accidents I got in as a teenager, and I think a lot of that was related to my attention span, to my ability to plan for what cars around me were doing. I wasn't really great at that with a lack of a fully formed frontal lobe. So as these kids start to get into puberty, there are a lot of hormonal changes that happen within the body, and those hormone changes actually also kind of flood the brain, and they actually change in some ways or affect neurotransmitter functioning. And so I already mentioned the change in GABA and the role that it plays, but the other thing that happens here is we tend to see a big spike in the development of psychopathology around the age of 12 or 13 when puberty really hits. And again, the reason being is those neurotransmitters oftentimes start to get triggered by the hormone changes in the body. And so as they, let's say, get out of whack, that's when we start to see psychopathology develop. So some factors that can influence brain development, things like heredity, your genetic composition obviously is going to, play a big role here. And then in terms of the environment, things like teratogens, so that can be alcohol, for example, prenatally, and nutrition prenatally, all of those things are going to impact the child. So the diagram you have here is of a child who was exposed to alcohol prenatally. So this is a child who has fetal alcohol spectrum disorder. And for some kids, there are physical abnormalities that we see that are kind of listed in the diagram here that tell us when a child has fetal alcohol syndrome. However, for many, many kids with fetal alcohol spectrum disorder, you would never, ever even know that they had this disorder based on their physical appearance. So a lot of times this is something that's not really encountered until later when parents start noticing a lot of behavioral symptoms. So that can play a big role in the way that a child's brain develops. And then finally, it's important to note these critical periods. So critical periods are limited age ranges in which particular kinds of environmental stimulation have the greatest and perhaps in some cases only impact. So a great example of that is learning other languages. So we think the critical period for this tends to be, kind of the younger you are, the easier it is to learn a second language, right? The older you are, especially after you kind of hit the teenage years, the harder and harder it is to learn other languages. And I think Ormond points out this idea of the L sound and R sound. So in some languages, they don't even have that sound. And when people go to learn English, it's sometimes hard for them to even hear or understand that sound because their brain has just never been exposed to it, and it's past this kind of critical period of development. So another thing that Ormrod mentions, and I'm not going to really spend a ton of time on this, but I wanted to clarify it for you because I don't think it's so clear in the book, is this idea of experience expectant or experience dependent influences on brain development. So experience expectant, the idea here is the elimination of synapses. So basically, there's certain skills that are going to be somewhat universal for everyone, right? Like crawling, for example. And almost any type of environment can foster these skills. So basically here, the idea is the cells that fire together begin to wire together, right? So we would expect this across experiences. Maybe that's a good way to think of experience expectant. Experience dependent, however, means that this is unique to particular cultural or social groups, and these skills only emerge when the environment nurtures them. So here we're talking about the addition of synapses based on experience in the environment. Hopefully that kind of clears up that concept for you, but definitely post in the discussion post for today if you're still struggling with the concept. Okay, so how pre-wired are we? And essentially what we know from research with infants in particular is that by 24 hours old, little tiny babies are able to discriminate between distance. At 1 to 2 days old, these babies have the ability to imitate an adult's facial expressions. And at 3 to 4 months of age, most babies start to show signs of surprise. So it does seem like there is kind of a built-in core knowledge about the physical world that babies are essentially born with. Now, that's not to say that all of these things are necessarily totally universal, right? But there is some evidence here from kind of the universality of the development of these skills that leads us to kind of believe there is something that's sort of pre-wired here. And what I'm going to show you now is the visual cliff experiment. So this is something that you'll see these babies all are kind of pre-wired to understand depth, right? And know when they're safe or not safe.
Emotion is a nonverbal language. Emotions reveal the cognition, the understanding of the baby. And furthermore, emotions are the nonverbal communication of the baby towards the parent and the parent towards the baby. Therefore, I thought that emotions were a royal road, one royal road, to the study of the baby's development.
In this study, babies between 9 and 12 months are brought into the lab and placed on a large plexiglass top table. Half of the table has a checkerboard pattern just underneath the surface, but halfway across is a visual cliff, which the baby can tell drops off steeply. The plexiglass top continues, so it's perfectly fine to proceed. But the baby isn't so sure, and this is a big drop for a baby just starting to crawl. She wants to get across to get the toy, but she's cautious and looks to the opposite end of the table where her mother is. The parent is instructed to smile or make a fear face.
If the mother is posing a fear face, the baby typically does not cross this stair-step downward, this modified visual cliff or visual step. On the other hand, if the mother poses a smile or somehow poses a nonverbal communication that is not prohibitive but encouraging, the child is much more likely to cross over to her. This particular study demonstrates the role of nonverbal communication in determining the child's behavior in uncertain contexts. The baby will, when they encounter something ambiguous, something uncertain, will typically look to the significant other, the mother, the father,
Okay, so not only did you see in this experiment that the baby kind of has an understanding here of depth, right, from this visual clip, but also what you saw is that babies are, for the most part, if they're typically developing, oftentimes pre-wired to kind of engage in almost this social referencing, right, to see if what they're doing is actually safe. So again, one of those things that we see very young. In terms of the implications, this is our last slide, I think. Again, synaptic pruning can actually be a good thing, right? We want this to happen, and it can be a function of learning and experience in the environment. So what we know is that enriched environments are critical to brain development. And then also, brain development continues throughout the lifespan. It doesn't just stop or we hit a ceiling at a certain point in adulthood. There's really no such thing as teaching to the left brain or the right brain. There's also no such thing as learning styles. This whole idea of auditory learning styles or visual learning styles has been disproven over and over and over again. We'll talk some more about that in this course, but essentially when people say that they're more of a visual learner or an auditory learner, it just essentially means they've been relying on those methods of learning maybe more heavily than others. But it's not that your brain is hardwired that way. So understand that. The other big thing here is that despite these critical periods that we see, the windows of opportunity remain cracked, as Ormond says. So in other words, it's not that you can't ever learn a language if it's outside of the critical period. It just might be more challenging for you. And finally, what we know is that brain research can help us understand learning theory, but it doesn't necessarily tell us what to teach or how best to teach it. And that's where we're going to start getting into learning theories that can actually help us with this. So the big theme here is that the environment is critical, and you see me mention this on several occasions. It really has a huge impact on the brain and the way that we learn. So we'll start out by talking about that in our next podcast.